Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine one of the toughest challenges in health, pancreatic cancer. The survival odds are just incredibly low.
Yeah, really grim.
Now, what if this enemy had a secret helper? Not like an outside force, but something woven right into its own support crew. We're diving into some really fascinating new research today. It's about a protein hiding inside the very cells that, well, actually help the tumor thrive.
And this could seriously reshape how we understand it, maybe even how we fight it.
Exactly.
So yeah, today's deep dive is all about this groundbreaking study. It was published in the Proceedings of the National Academy of Sciences, PNAS.
Right.
And it's identified this, well, totally new function for a protein called Galectin-1.
What Galectin-1, okay.
We kind of knew it was hanging around pancreatic tumors, specifically in these non-cancer cells, fibroblasts. They make up this thing called the stroma, the tumor's environment.
Wait, the support structure.
But what the study found is that Galectin-1, it's got the secret life inside those fibroblasts.
Okay, interesting. And we're pulling this apart using the scientific paper itself, primarily.
Yeah, the PNAS paper, and also looking at a health news report that covered the research, just to get a, full picture.
Great. So our mission for this deep dive,
Yeah.
let's really get our heads around this new Galectin-1 finding. What does it mean for how aggressive pancreatic cancer is? And crucially, could this lead to new ways to treat it?
Let's do it. So pancreatic cancer, as you said, very aggressive, low survival rates. That hasn't changed much, sadly.
No.
And a big part of why is this tumor microenvironment. It's this whole complex ecosystem around the cancer cells.
The neighborhood the tumor lives in.
Exactly. And the stroma is a huge part of that. It's not just cancer cells, it's proteins, other cell types, and fibroblasts are really key players there.
Fibroblasts, sometimes called pancreatic stellate cells, right?
That's them. And they're not passive. They actively help the tumor. They support its growth. They even help it resist drugs.
Okay, so we knew these fibroblasts were involved, and we knew they were released of this Galectin-1 protein, and that protein helped the tumor from the outside.
Correct. That was the established picture. Secreted Galectin-1 is pro-tumor.
But this new work has thrown a curveball, you said. Found it inside. What's it doing in there?
This is the really cool part. They showed Galectin-1 is also inside these fibroblasts. Specifically, it's in the nucleus.
The cell's command center.
Precisely. And in the nucleus, it's doing something completely different, something vital. It's regulating genes, deciding which genes get turned on or off.
Wow. Okay, so a totally unknown role inside the cell.
Totally unknown until now.
So this protein is like moonlighting, working outside and inside these helper cells.
Kind of, yeah. A double agent, almost.
How does it actually control those genes from inside the nucleus?
So the mechanism they identified is epigenetic control.
Epigenetic. Okay, what does that mean exactly?
It means it influences which genes are active without actually changing the underlying DNA code itself.
Ah.
Think of it like putting sticky notes on a blueprint. It doesn't change the drawing, but it tells the builders which parts to focus on or ignore. It can switch genes on or off, or maybe just, turn the volume up or down on how much protein a gene makes.
Right, like a dimmer switch for genes. So it's regulating specific genes in these fibroblasts. Did they find out which genes?
They did, and this is a really critical connection. They found that nuclear Galectin-1 controls the activity of the Kras gene.
Kras, that name rings a bell. Isn't that a gene that's often mutated in the actual cancer cells?
Exactly. Kras mutations are found in the tumor cells themselves in the vast majority of patients, maybe 90%. It's a huge driver of the cancer.
Wow.
But here's the twist. This research shows Galectin-1 is controlling the normal, the non-mutated version of Kras, but inside the fibroblasts.
Inside the helper cells, not the cancer cells.
Right. Inside those supportive fibroblast cells. And even though it's the normal version of the gene, controlling its activity in these helper cells still seriously boosts the tumor's growth and aggressiveness.
That's wild. So the tumor is somehow manipulating its own support network using their own genes.
It really looks that way. It's like it's hijacking the fibroblasts' internal machinery via this Galectin-1 protein to make the fibroblasts better helpers for the tumor.
So even if the main problem starts with a KRAS mutation in the cancer cell, This effect in the fibroblasts adds fuel to the fire.
Precisely. It's another layer contributing to how bad this cancer can be.
Okay, how did the researchers actually figure this out, this hidden role inside the nucleus?
Well, they used a few different approaches. They looked directly at tissue samples from pancreatic cancer patients.
Real patient tissue.
Yeah, that let them actually see the Galectin-1 inside the fibroblast nuclei. They could pinpoint its location.
Okay.
Then they also did experiments in the lab using human fibroblast cell lines.
So controlled experiments.
Right. And in the lab, they could, manipulate things. They reduced the amount of Galectin-1 in the fibroblasts, or they specifically blocked the Kras gene in those cells.
And what happened?
When they did that, the fibroblasts became much less active. They stopped supporting the tumor cells as much. They were essentially deactivated.
Any other techniques?
Yes, they use something pretty sophisticated called chromatin immunoprecipitation sequencing, CHIP-seq, they sometimes call it.
Okay. Sounds complex.
It basically lets you map out exactly where proteins bind to DNA, so they could see precisely where Galectin-1 was latching on to the fibroblast's genome, which genes it was physically interacting with.
Got it. So patient samples, cell lines, and this detailed mapping technique all pointed to the same thing.
Exactly, a multi-pronged confirmation.
This really does change the picture of how the tumor and its stroma, its environment, talk to each other.
Absolutely. It's not just the fibroblasts pumping out helpful stuff from the outside.
No, it seems much more intimate than that.
Yeah, it gives us a much deeper insight into that communication. It shows these fibroblasts aren't just, passive bystanders. They're being actively co-opted from the inside by this nuclear Galectin-1. They're being pushed to become better collaborators for the cancer.
So if our understanding is shifting, what does this mean for treatments? Are there new angles here?
This is where it gets potentially very exciting. Most efforts to target Galectin-1 so far have focused on blocking the protein after it's been secreted.
The stuff floating around outside the cells?
Right, trying to mop it up or stop it from binding to things externally. But this finding, it strongly suggests that's only half the story. Future treatments probably need to also deal with the Galectin-1 that's working inside the fibroblast nuclei.
So we might need drugs that can actually penetrate these fibroblast cells, get inside, and stop Galectin-1 there.
That's the thinking, yeah. Developing new types of inhibitors drugs that can work within the fibroblasts, specifically targeting that nuclear function.
It makes sense. And tackling it both inside and out, that could be a more powerful strategy?
You'd think so. Because Galectin-1, even the external stuff, is involved in other bad things for the patient.
Like what?
Like helping the tumor build new blood vessels, angiogenesis, which feeds it.
Oh, okay.
And it's also implicated in helping the tumor resist immunotherapy treatments, trying to get the patient's own immune system to attack the cancer.
Right. So blocking Galectin-1 everywhere could hit the tumor on multiple fronts.
That's the hope. A more comprehensive attack could have, multiple anti-tumor effects.
Now, we always need to be careful with new research. What are the caveats here, the limitations?
Well, the big one is that this is still preclinical.
Meaning not tested in human trials yet.
Exactly. The findings are based on those patient tissue analyses and the lab cell line experiments. We need more research to confirm it all works in, say, animal models first.
Living organisms.
Right. And then, eventually, carefully designed human clinical trials. That's a long road from the lab bench to the bedside.
Always is. What about the complexity of the tumor itself? Is targeting just Galectin-1 going to be enough?
That's another key point. The tumor microenvironment is incredibly complicated. So many different cells, so many signals flying around. Galectin-1 is clearly important based on this study, but targeting it alone, probably not a magic bullet. It's much more likely that effective future treatments will involve combination therapies.
So hitting Galectin-1 plus maybe something else.
Yeah, hitting Galectin-1 perhaps both inside and outside the fibroblasts, along with targeting other pathways or other cell types in that microenvironment. That's usually how cancer therapy evolves.
Makes sense. This work must be generating some buzz among the experts. What are they saying?
Definitely. Dr. Pilar Navarro, who leads the research group behind this, she really emphasized how critical the stroma is for pancreatic cancer's aggressiveness and just how significant finding this nuclear function of Galectin-1 is.
Yeah, it opens a new chapter.
And Dr. Nus Martinez-Bosch, another researcher involved, she specifically pointed out how this shifts the therapeutic focus. We now have to think about blocking it inside the cell, not just outside.
A clear implication for drug development.
Then there's Dr. Judith Veniksa, the study's lead author. She highlighted how their results confirm Galectin-1's crucial role inside the nucleus, controlling genes that are key for how these fibroblasts behave and support the tumor.
And looking ahead.
Dr. Gabriel Rabinovich talked about next steps, exploring combinations, like we just discussed, therapies targeting both the external and internal Galectin-1, and he also stressed its relevance to those other processes, like blood vessel formation and immunotherapy resistance.
So connecting it to the bigger picture.
Right. And even the PNAS paper itself, in its significance section, points out how these findings offer a really unique view on how the tumor environment works dynamically and the potential to reprogram these tumor-helping fibroblasts by targeting Galectin-1.
Reprogramming the helpers. That's a powerful idea.
It really is.
Okay, so let's wrap this up. The big takeaway here is this unexpected discovery. Galectin-1 isn't just outside fibroblast cells and pancreatic tumors, it's also inside, in the nucleus.
Right. And it's actively working there, controlling key genes like Kras to essentially activate these fibroblasts, making them better supporters of the cancer.
This fundamentally changes our understanding and points towards needing new therapeutic strategies, potentially drugs that can get inside these fibroblasts.
Targeting both the internal and external actions of Galectin-1.
So here's a final thought to chew on. What if we could effectively silence that inner voice, that nuclear Galectin-1 within the tumor's own support system?
Could we change the whole nature of this disease?
Could we fundamentally weaken this aggressive cancer, maybe turn the tide and really improve outcomes for patients? It certainly opens up some very exciting new avenues for researchers to explore.
Definitely a space to watch.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.